In this lab, we will talk about some aspects of the sensory perception. We're going to discuss some tests for different sensations. Since we cannot perform that, we'll have to kind of make stuff up. So the first sensation that we're going to be talking about is the sensation of taste and sensation of smell. We'll look at these two together because they are inextricably linked. The scientific term for sense of taste is gustation. Scientific term for smell is olfaction. In both cases, chemoreceptors sense chemicals that are dissolved in mucus in the nose for olfaction or saliva in the mouth for gustation. Your tongue has a lot of papillae. There are different papillae, fungiform papillae, valvulate papillae, filiform papillae. And inside of these papillae are multiple taste buds. In these taste buds, you're going to find receptor cells, and the gustatory hair with the receptors of these receptor cells are sticking out on the surface of your tongue and convey the sense of taste. You know, they sense the chemicals and transduce that interaction of a chemical with the receptor into the nerve signal which they transmit to the brain. So there are 5 different tastes. It's sweet, sour, salty, bitter, and umami. So sweet, sour, salty, and bitter, usually, easy to understand. Umami is the taste of a protein, savory taste. So smell as well as taste both are necessary to fully appreciate the food. Okay. So in olfaction, the epithelium that senses the smells is located in the roof of the nasal cavity. It consists of multiple olfactory receptor cells, olfactory neurons so-called, and the cilia of these olfactory neurons is embedded in mucus. Odorant molecules will be dissolved in mucus. They will interact with the receptors on the olfactory neurons, and these olfactory neurons will convert that interaction into the nerve signals, which will go through the olfactory nerve to the brain. Now, relationship between the taste and smell. That's actually a pretty cool story. We're going to kind of, play it. Okay, so I'm going to tell you that there is a red. So what we usually do in this exercise is pretty cool. So one person is blindfolded, and this person tries to taste the lollipops of different types with the nose closed like this or not. So we're going to hypothetically red, green, and yellow. Okay. And the red one without the smell, let's say it's going to be watermelon. For green without the smell, it's going to be apple. And for yellow, it's going to be cherry. Well, it turns out, if you open it up and red becomes apple, green becomes watermelon, and yellow becomes lemon. So you see that those are actual true tastes that we perceive. So without the smell, taste dramatically changes. So based on our results, is smell required for an accurate perception of taste? Yes, of course. Okay. Initial hypothesis. We didn't really formulate it, but if your initial hypothesis was that smell is essential for taste, then yes, it was supported. Human bias, we took the steps of blindfolding participants during that experiment. Potential sources of error is that the measurement is not quantitative, it's qualitative, and people have different taste perception and different sensitivity to the smell. In your skin, you're going to find multiple receptors as well. Receptors like nerve endings, like this nociceptor, which senses the pain. Masonry corpuscles that sense mechanical stimulation like vibration. Ruffini corpuscles that are deeper, and bulbous corpuscles that are very, very deep. So they sense a pretty significant pressure. You're going to find hair root receptors, Merkel discs. So your skin is incredibly sensitive. Now, types of receptors that can be found in your skin are nociceptors that are activated by pain, thermoreceptors activated by temperature, you can sense warm and cold, and mechanoreceptors that will sense pressure, vibration, touch, stuff like that. So in this exercise, it's actually pretty interesting. It's called 2-point discrimination test. We'll get back to that picture, but basically what we try to see is where the density of the receptors is highest. Because you have to appreciate, if the density of the receptors is really high, then each receptor will cover a very small area. And you will be able to discriminate between two mechanical stimuli on the skin much better. Now, how are we going to, and we're going to say that our hypothesis will be that the highest density of sensory receptors will be found on the fingers. So the person will be blindfolded, and then the investigator will have the calipers, okay? And using these calipers, we will move the ends of the caliper, the tips of the calipers, closer to each other. Closer and closer and closer. And then when the person stops sensing two tips and start to sense only one caliper tip, perceive them as one, we say at this point person cannot discriminate between two different points on the skin. So on the fingertip, let's say it's going to be 5 millimeters. Okay, for forearm, let's say it's going to be 17 millimeters, and it kind of reflects. Back of the neck is 8, and cuff, it's going to be 23. So you can see that, and those are pretty close numbers. You can see that for the fingertip, fingertip is the most sensitive. Okay, forearm is much less. Back of the neck is an incredibly sensitive area as well. And cuff is not sensitive at all. So which part of the body show the greatest sensitivity? Fingertip. Least sensitivity, cuff. Okay. Initial hypothesis, yes, it was supported. Minimization of human bias, blindfold. Potential sources of error, well, we all have different skin, different sensitivity on the skin, thickness of the skin. There can be some differences in terms of, where you measure which finger, which part of the forearm. So yeah, you can standardize this. Now, hearing. So sound is by pinna, the external ear. It focuses the sound into the external acoustic meatus, the auditory canal, where sound vibrates the eardrum, which in turn vibrates 3 auditory ossicles, malleus, incus, and stapes. Stapes transmits vibrations onto the oval window of cochlea, and that leads to the vibrations of the fluid inside of the cochlea. And these fluid vibrations, they move hair cells in the organ of Corti, and that transduces the sound into the nerve impulses which reach the brain via vestibulocochlear nerve. So we're going to label some pinna, it's right here, external auditory canal, tympanic membrane, malleus, incus, stapes. Oval window is right here under the stapes. Vestibule. Vestibule is in between the cochlea and semicircular canals. Semicircular canals. Cochlea. Vestibule and semicircular canals are responsible for the sense of balance. That's vestibulocochlear nerve, and that is eustachian or pharyngotympanic tube, which equalizes the pressure between the middle ear and the outside world. So, tympanic membrane and the malleus. That is the tympanic membrane, and that is the malleus. They are in the middle ear. When malleus vibrates, incus and stapes vibrate as well. Now, A is cochlea. B is the tube that connects the middle ear to the throat, is pharyngotympanic tube. Semicircular canals are involved in the what's called dynamic equilibrium, sensing the rotational movements of the head. Hearing imbalance, since in hearing imbalance sensation, receptors essentially detect the movement of liquid, those are mechanoreceptors. Now this, not much, otoscope is used to examine the ear. You can see some pathologies here. So normal ear with a perforation of the tympanic membrane, ear with inflammation, acute otitis medium, and ear with accumulation of inflammatory fluid with effusion. So this is the otoscope. Basically, I think everybody went through this exam, and it's pretty neat, but unfortunately we cannot do this otoscopy here. So review questions for healthy tympanic membrane. Why would health professionals be concerned if a patient's tympanic membrane red, yellow, or opaque? Because it means that it is inflamed and there is fluid accumulating behind the membrane in the inner ear. Hearing definitely would be affected by the middle ear effusion because the fluid would inhibit the normal sound transmission. So, sound localization. Unfortunately, we cannot do this lab as well, but I want to just exemplify. So when we have two ears and ability to localize the sound, we have it specifically because we are binaural. We have binaural hearing. Okay? So if you would blindfold someone and will make sounds for that particular person from different locations, the person will be able to point at where the sound is coming from. So sound localization doesn't really require the sense of vision, because due to the binaural hearing, you can localize the sound. Our initial hypothesis would be that, you can localize the sound being blindfolded, and it would be confirmed. If we would put an earplug into one of the ears, then what do you think the results would be? You're absolutely right. It would affect the results of the localization test, because instead of normal binaural hearing with both ears, hearing from one ear would be diminished. Sources of error, well, subjectivity of the people who do the experiment. Now, speaking of vision, okay, vision is done by an eye. You can see typical structure of an eye here. So the cornea, the sclera, the white of an eye, they form fibrous layer. You can see lens. So cornea is right here, I'm sorry. You can see lens. And then That's your iris with an opening that's called pupil. This is ciliary body. It consists of ciliary process and ciliary muscle choroid that supplies the blood to the retina and retina. Retina is the vision part of an eye. Retina is the nervous part of an eye. It contains photoreceptors. Now the vitreous chamber, the back part of an eye, is filled with vitreous humor, and the anterior chamber here is filled with the aqueous humor. So this image shows you the different structures of an eye, from lens to nervous layer to vitreous chamber. We're going to jump directly to how we see the objects. Interestingly enough, the objects that we see are flipped upside down and left to right. Another interesting thing is that sensory information that is collected by the retina is channeled to the brain by the optic nerve. So this part right here where optic nerve enters retina is called a blind spot or optic disc. That's the place where there are no photoreceptors. But our brain is able to, first of all, flip them back and, downside, from upside down to normal, from flipped left to right to normal. And it also is able to compensate, our brain is able to compensate for no photoreceptors on the optic disc. So let's talk about main structure. So cornea, right here, sclera is the white part. Pupil, kind of should be somewhere here. Iris, that's the muscle called iris. Ciliary muscles are right here. Lens, that would be lens, and that would be lens hanging in there. Choroid, okay, that's the brown layer. Retina is the yellow layer. Optic disc would be here and here. Anterior cavity, would be this part, and vitreous chamber would be this part. So sclera, iris, and the pupil. Well, pupil is the dark spot. Sclera is white of an eye. Iris is that gray area around the pupil. Diameter of a pupil is adjusted by iris. Iris is two muscles, circular sphincter pupillae and radial dilator pupillae, that will constrict. When sphincter constricts, it makes pupil smaller. When dilator constricts, it makes it larger. So the higher the intensity of light, the narrower the pupil. Okay, lens here. Retina is that dark black colored, well, and the opaque part of the eye. Vitreous jelly just inside of the eye. Function of the lens is to focus the image of an object on the retina, and photoreceptors are located within the retina. Fortunately, we don't have the otoscope opportunity to examine the eye, just show you. So this is the retinal detachment. Usually it is a result of a severe blow to the head. And here you can see macular degeneration. It's the progressive degenerative condition characterized by the loss of vision with age. Now, this is the ophthalmoscope, a small ophthalmoscope. So that's the window through which you view, and there is a light that is produced through that window so you can see better. So you basically shine the light into an eye and you can see an optic disc and blood vessels at the retinal circulation. I mean, we cannot do anything. Near point accommodation test. So that's the closest when somebody can see the words on the pencil sharply. It will be different for people with different types of vision. For instance, for a person with, we're not going to get any data here, but for a person with myopia, short-sightedness, of course, the near point accommodation distance will be smaller, so the pencil will be closer. For a person with far-sightedness, hyperopia, good distant vision, the near point accommodation distance will be farther. So we cannot do this. Yeah, age does seem to play a role in near point accommodation. Usually with age, people become more far-sighted. So near point accommodation distance will be larger in older individuals. So 90-year-old man who is not wearing reading glasses compared to, say, a 20-year-old, his test results would be longer distance. Same 90-year-old man, he's wearing reading glasses. If glasses are correct, his results will be the same as yours if you have normal vision. And finally, the review questions for the lab. So photoreceptor, okay, photoreceptor, vision, nociceptors, pain, chemoreceptors, taste and smell, mechanoreceptors, touch, hear, and balance. Tympanic membrane, here, malleus you can kind of see in the back. When sound waves enter the ear, tympanic membrane vibrates. Sense receptors for hearing. Well, you can say cochlea, and inside of the cochlea it's organ of Corti. That's where the sense receptors for hearing are. Sound information is transmitted to the brain via vestibulocochlear nerve. Skin modal, here's epidermis, here's dermis. Blood vessels and the majority of sense receptors are located in dermis. Skin varies in its sensitivity to touch because the density of the receptors, sensory receptors, is different in different parts of the skin. So auditory ossicles, D and E, they're located in the middle ear. Receptors for balance are located in G, the semicircular canals, as well as vestibule. Name of the structure H is cochlea. Photoreceptors are located within the retina of an eye. The optic disc, the image below is here. In this part of the retina you can find an optic nerve. Sound localization is ability to identify where the sound is coming from without visual clues. Ears can localize sound waves because there are two of them, and they detect small differences in timing when the sound arrives, in the loudness, and so on and so forth. Iris is labeled none, so actually they failed to label iris. So this is iris. B is a pupil. Optic disc is labeled F. The function of the structure C, that's lens, is to accommodate the vision, adjust the vision for the proper distance, to focus the image on the retina.